Hello there, this is a recording about cell organelles, what they are and what they do. Now, one of the things that'll happen every now and then is that Mr. Kabelki and I won't present a presentation on PowerPoint. We'll actually run this as a movie and we'll let you just work through the movie and you can use it as you see fit. So the syllabus dot point that we're looking at this time is to process information. So basically what this means, you'll be given some pictures and you should be skilled sufficiently to be able to identify the organelles and tell us what they are. And all of the organelles that you can see listed here will be the ones that we make sure that we have covered for you. Now there are a few key points here that you should be able to work out by using the materials that are supplied with you in class. First one is that you should be able to recognise organelles as shown under the light microscope, and that'll be the type of microscope that we'll be working with. We'll be recognising organelles as shown by the electron microscope, and these will be the ones that will be given to you as second-hand data. All right. Also know which types of cells have them. In particular, you should be able to work out the distinction between a plant and an animal cell depending on what organelles you can see. And this is something that was even talked about in Year 7, certainly would have been talked about last year in Year 10. And then finally, you need to be able to describe the function of all of these organelles that we'll present to you. Now this shouldn't pose too big a problem because last year in Year 10, you did a number of these already. Now before we continue, one of the things that I'd just like to point out to you is that there are two main cell types. There are what are called prokaryotic cells, and these are found as bacteria. And what you have here is you have circular DNA. It's not in a nucleus that you're familiar with from junior school. Okay, it's basically called naked DNA. It's found distributed throughout the cell. Okay, you will see that they do have things like membranes and cell walls, something that you should be familiar with already. And also, bacterial cells also have ribosomes. Now, eukaryotic cells not only are animal cells, but they're also plant cells as well. And the big distinguishing feature between a eukaryotic cell is that it has the DNA in a nucleus which is actually bound up by a membrane, and that's sort of what I'm trying to circle around on the diagram here. So that's what we would call the nuclear membrane. The other thing too is it has lots and lots of different types of organelles, and all of these organelles, with the exception of the ribosomes, are also bound up with membranes as well. So lots and lots of membranes, not only just encircling the cell, but also inside a eukaryotic cell. So just got a couple of examples of cells as seen by a light microscope here. The one on the left, this one here, I'll just mark with the X, is a cheek cell. Okay, you probably wouldn't have done this because of health risks and health issues, but basically what you can see here is you can see the nucleus and you can certainly see the membrane around the outside. There's a whole heap of grainy stuff in the middle. That is the cytoplasm, and that's pretty much all you can see with an animal cell under the light microscope. On the right hand side, however, we actually have some onion skin cells. Once again, something you would have seen lots of. You can probably see the little nucleuses all over the place here. I'll just put little lines where you can see them, just next to a few. Okay, and then on top of that, you can also see very distinctly where the cells start and finish. And the reason you can see that is because that's the cell wall. Cell membranes are notoriously tricky to see, and you can't actually see them with a light microscope. What you're actually seeing is the edge of the cell, and you go, oh, that's supposed to be where the membrane is. So these are a couple of things that you're already familiar with. Now this is another example of a plant cell where we've got the leaf in section here, so cut sideways to a leaf, and what we can see the nucleus over here, so I've just filled that on in where it was. We can also see the little green spots around the sides. Those are obviously chloroplasts, and in the middle we have this large vacuole, and we can also see the cell wall. Okay, it's pretty distinct here. Now once again, these are the types of things that you can see in plant cells. All right, obviously with a plant cell, chloroplasts and vacuoles are usually pretty easy to pick up, and it's a nice distinguishing feature to be able to tell the difference between a plant and an animal cell. So I guess the take-home message from all of this is that there are structures inside cells, and as you're probably already familiar with when we've been talking about cells to date, we call these organelles, which just means little organs. And like the organs in our body which carry out specific functions, organelles do much the same thing. And as I was saying before, all organelles have a membrane which encloses them from the rest of the cell. So all organelles are bound up in their own little membranes. And the other thing is that the only organelle that doesn't have a membrane is going to be the ribosome. We'll talk about the function of the ribosomes a bit later. Now obviously one of the things about electron micrographs by comparison to the light microscope images that you will see is that things are much bigger and you can bring the organelles up into much sharper focus and see things that you could possibly never see using a light microscope. So the first thing that we've got here is the nucleus, and obviously the nucleus is the place where the genetic information of the cell is being stored. And you can see around the outside, that's the nuclear membrane. I'm sort of going around part of it here with my little highlighting tool. All right, and then we have this dark structure in here, which I'm circling in red, and that's called the nucleolus. Now, if you were to liken the nucleus to a library, the nucleolus would be the books in the library that are being read at any particular time. So what's actually happening is the genetic information that is stored in the cell, the part of it which is being used by the cell to help keep its functions ticking over, is currently happening in the nucleolus. Now this particular slide is showing a plant cell, and you can see here that the structure that has been labelled for you is the cell wall. The cell wall provides structure and rigidity to plant cells and is obviously only found in plant cells. So this is another image which has been taken from a plant cell, and we can see here we've got the cell wall on the edge here, which is quite thick, but the structure of interest in this particular case is the chloroplast. Now if you are trying to identify chloroplasts using an electron micrograph, the thing to look for are these stacks here. Okay, so you've got these little folds which are all stacked on top of each other. They look like a bit like a stack of coins. These are called grana. You're not expected to know that, but it's just a nice identifying feature. And one of the things which I'm sure you know the chloroplast does is it performs photosynthesis, again for the plant cell. Again, chloroplasts are unique to plant cells. So here's another picture of a plant cell, and you can see around the outside there are multiple chloroplasts there, and hopefully you can actually see those little stacks that I was talking about as a distinguishing feature. Now this particular organelle is the mitochondria that we're going to look at next. Now the mitochondria is often called the powerhouse of the cell. It is where the cell derives its energy from. Now mitochondria are found both in animal and plant cells. You might be wondering, why do plant cells need mitochondria when they're photosynthesizing all the time? But the rub is that plants actually need to break down that glucose again and take it to the various parts of the cell where they need it. And on top of that, plants don't photosynthesize in an absence of light, but they still need energy to keep their functions ticking over. So that's the reason why. Now, one of the things about mitochondria is that they can be very thin and sausage-like to being almost circular in their shape. However, to make life easier for you guys, we will probably just give you long, thin examples that you can look at and identify in future tasks. So this is pretty much a typical mitochondria that you would find in an electron micrograph. Now the mitochondria is that entire sort of sausage-shaped structure there, and the distinguishing feature for this is that it has these finger-like projections running inside it like these. These are called cristae. You don't need to know the name of those, just to help you identify these things. Now the next structure that we've got here is called the endoplasmic reticulum. Now I'm just going to highlight where it is. So it's basically these channels which I'm trying very badly to draw in blue here, okay? These things are the endoplasmic reticulum. Now they have a number of different functions. The primary functions that you need to be aware of is that the endoplasmic reticulum actually helps a cell transport substances from one part of a cell to another, and they are sometimes involved in the synthesis of fats. Now there are two types of endoplasmic reticulum. On the previous one where I was filling it in, you would have noticed that there was lots and lots of little dots on it. That's what they call rough endoplasmic reticulum, and that's because there are lots of ribosomes stuck on there. The other type of endoplasmic reticulum that you can have is over in this part of the slide here, and you can see you've got those long channels running through the cell here, but there's none of those ribosomes. So this particular endoplasmic reticulum is called smooth endoplasmic reticulum. Now the next organelles that we have here, there's two of them. Okay, we've got a electron micrograph at the top, and we've got a diagrammatic representation of it down the bottom. The Golgi body is this large part in the center of the slide here, and then you'll also see, and I'll just change color here, you'll see these little bubble structures coming off to the sides. These are called vesicles. Now the Golgi body's role is to package materials so that they can be exported or secreted from the cell. The vesicles or the lysosomes, now the names can be used interchangeably. It depends which type of text or internet site you're looking at. The vesicles or the lysosomes, those are the things which actually transport them to the surface of the cell and then release them. Now we're back to the plant cells again. So this is a structure which is distinct for plant cells, and this is the vacuole. This stores water, minerals and other substances. Vacuoles do exist in animal cells, but they are very, very tiny and very, very hard to find even with an electron micrograph. So from our point of view, we'll just say that vacuoles are characteristic of plant cells. Alright, the next structure that we have here are ribosomes, and these will appear as small black dots within the cell. Sometimes they will be attached to endoplasmic reticulum, sometimes they won't be. So we can see here this particular section has the ribosomes on endoplasmic reticulum. Down here the ribosomes are free and just in the cytoplasm. Now the next structure that we've got here to discuss is the cell membrane. Now the cell membrane does two things. First, it actually acts as a barrier for the inside of the cell and the external environment. But on top of that, the membrane allows for the selective transport of substances while excluding others. And that's a very, very important role in all cells, because we don't want everything to come into the cells, and sometimes we don't want everything to leave the cells as well. So very, very important, and will be the subject of a bit of discussion later on. Now the cell membrane is not very easily seen, and it is nowhere near as obvious as the plant cell wall is. And often basically all you can do is you can work out where the cell membrane is just because it is at the end of the cell. So here we've got a plant cell and we can see the cell wall just here, but down in here we can see where the membrane is, where it's peeled away from the cell wall. Now in this particular slide, the cell membranes are easy to see, or where the cell membrane should be is easy to see, because the two cells are separated and we can see through the gap just indicated here by the red line. So the two edges here are where the cell membranes are. Now the final organelle that we need to go through is the cytoplasm. Now the cytoplasm is basically everything inside the cell membrane. Whoops, let's get rid of that. Everything inside the cell membrane, between the membrane and the nucleus. So all of this in here is cytoplasm, and that includes any organelles which are sitting inside there as well. So the cytoplasm is basically for want of a better description, a jelly-like substance containing dissolved things, minerals, proteins and so on, but it also has the organelle suspended in it. Now if I had a space there where there were no organelles, we would call that cytosol. However, we'll just concentrate on cytoplasm. I only mention cytosol because some textbooks do. Now, I can't speak for Mr. Kabelki's class, but one of the things that I always like to do is I like to encourage my students to write some notes. And this particular movie is no different. So what I'd like you to do is you construct a table with the following column heading. So it'll be the name of the organelle, the type of cell it is found in, whether it's visible under light or electron microscope, and its function. And obviously if it is visible under a light microscope, it must be visible under an electron microscope. So we've got a 4-column table which you need to complete. So I'll let you get started on that. On top of all of that, there are the usual quizzes and little homework tasks that we will get you to submit on Moodle as well. But this pretty much wraps up the first section of this particular topic. So congratulations.